Okay, session 17. So this is all about axon functions and different kinds of neurons and how they fire, more of the physiology of it. I feel like last couple sessions we did were about, were about the names and the structures, and this will be more physiology heavy. Okay. All right, so the axon is a conducting region of the neuron, right? So where's my little doodad? This, right, the axon is the conducting region of the neuron. That's where our action potential, which is what this little wave is, runs down, right? So say we get, let's see, where's the thing that allows me to draw? Cool. So say we get an impulse right here from another neuron. It'll travel down these dendrites through the perikaryon or the soma, right? That's what this is. That's just, we'll look at that later. It's like on a graph. It would be like negative 70 millivolts, depolarized, action potentials up here, right? We have an axon potential that's fired, and then we go back down to negative 70 millivolts. Anyway, which is what this is. So we go down the axon, and then we transmit this down here to the axon terminals, which maybe will be at a muscle, an organ, a gland, an effector organ, anything like that, because we're just sending the signal, right? It's a conducting region. We're conducting electrical impulses, which tell us what to do, which tells us to blink, tells us to move our eyes, tells us to do anything we're supposed to do. That's how we communicate. Okay. And good. I'm gonna just leave this doodad up. Okay, so it generates a nerve impulse that travels along the axolemma, right? We said a sarcolemma was the membrane, the cell membrane of a muscle fiber. So the axolemma is the cell membrane of a neuron, okay? So it transmits along the axolemma to the axon terminal, right? To the end. This is an axon terminal, right? The terminal is where we secrete that neurotransmitter, right? If you remember in the muscle video, we saw that acetylcholine come out of the axon terminal. Okay, remember there were these little secretory vesicles, and inside of them there were the triangles, and that was acetylcholine, and those came out of here because we had an action potential. Sorry, this is challenging. An axon potential that came down this, which told these and the acetylcholine or whatever hormone it is to come out, right, and to signal whatever cell. They're all my drawings. Oh, that's easier. Okay. Okay, and what can it do? It can excite or inhibit a neuron, right? So it can either tell it to start or stop or continue or get hard or increase or decrease. Okay, it communicates with many different neurons. It communicates with many different neurons at the same time, right? So that's why we have so many dendrites and we have so many axon terminals, because we can send one message down lots of axons and it will transmit everywhere, right? So all of these, right, we send a message here and here and here and here and here and here, okay? Okay, so a myelin sheath, right, so they're composed of a myelin sheath, which is a white protein lipid structure, okay? So protein and lipids, so it has proteins and it has fat in it, and it's a structure substance, right? And its function is to protect and electrically insulate and increases the speed of the nerve impulse transmission. That's where our lipid comes in, okay? Our lipid increases the speed of the nerve transmission because it allows us to have these things called nodes of Ranvier and those myelin sheaths, which we'll look at in a minute, so do not be afraid, that allow us to have that electrical signal jump, okay? So it's composed of a myelin sheath, right, which protects and it increases the speed of transmission, right? We want to make sure, remember that in your nervous system, that's our fast acting response system. And so we want to make sure that we're doing it fast. So we want to make sure we're getting the message, right? So it's insulated and it's going to get there and that it happens quickly, okay? So there are fibers that are myelinated and there are fibers that are non-myelinated. Where do I want this? There are fibers that are myelinated and fibers that are non-myelinated. So myelinated pretty much just means they have this myelin sheath. They have this white protein lipid substance that protects and insulates and increases the speed. So if it's a myelinated fiber, right, it's surrounded by a long axon that has a myelin sheath. And so it's going to do these things. It's going to have protection, insulation, and it's going to have an increased speed of transmission. However, the non-myelinated fibers will not, okay? So these do not contain a sheath and they happen more slowly, right? Our electrical impulse goes more slowly. Okay, so myelination is formed in Schwann cells, right? So if you don't remember what a Schwann cell is, go back to the last session or to, where is it? I want you to see this video, but also go away. But I want you to look at this video, okay? Divisions of the nervous system, and it's in that helpful for video or for exam right sorry. When you're applying to med school, you get a lot of med school ads. I find it interesting how like ads are targeted to everyone. Anyway, go watch like Jeopardy at like 9 a.m. and you'll see some very different ads. Anyway, this video, where's the frontal breakdown? Okay, it gives you all the divisions of the nervous system, and it talks about what kinds of cells and things comprise each part, right? So it's important, as well as this video where we talk about each kind of cell in each place, right? And so it's important to make sure that you can differentiate different kinds of cells and their function based on whether they're in the central or the peripheral nervous system, okay? So myelination is formed in Schwann cells, and its function is to wrap around the axons in a jelly roll fashion. Okay, so, which pretty much looks like, give me a neuron, come on. Okay, that doesn't write myelination on it. Oh, there it is, there's one. Also, this guy is awesome. If I'm being honest, I watch him for a lot of like physics and MCAT stuff, and I think he's really good. So, okay, right? So these, right, these little blue pieces are the myelin sheaths. Those are those wrappings of the myelinations that are formed by Schwann cells, which allows us to have an electrically insulated and protecting fiber, and it also allows us to increase the speed of impulse. Okay, so that's what these look like. Okay. And so then they form these nodes of Ranvier, which is the thing I said earlier, right? They're gaps between the Schwann cells, right? Or those little bubble jelly roll things that acts as axon collaterals and can emerge and allow us to have a faster conjunction. So, right? So we get an axon, we get a thing, okay? And then the action potential, right? It's chilling, it's going down, right? And then it goes to here, right? But instead it goes like this. It jumps from here to here to here. It jumps from the parts that are insulated to the parts that are non-insulated, right? So it jumps here and here. So it goes faster, okay? Clear all my drawings, okay. Okay, so those are the nodes of Ranvier. Those are super, super important. All right, so myelinating sheaths in the central nervous system, right, are formed by oligodendrocytes, right, which are similar to Schwann cells, which might mean that Schwann cells are in the peripheral nervous system and oligodendrocytes are in the central nervous system, right? So they're formed by oligodendrocytes, but not the whole cell, okay? We don't wrap the whole cell, we just wrap that axon, right? So its function is to wrap axons, okay? So we have white and we have gray matter when we're talking about the central nervous system, right, which is our brain and our spinal cord. So white matter is a dense collection of myelinated fibers and fiber tracts. So it makes sense that the white matter is going to be lots of myelinated fibers, right, lots of lipid and protein heavy wrapped fibers, right, which gives it that white character, okay? And then gray matter is mostly cell bodies, right, or non-myelinated fibers. Okay, so picture this. Whatever we're doing, fibers, axons, all that jazz is originally gray, okay? Which is why the gray matter is non-myelinated. However, when we myelinate something, right, when we give it that axon wrapping, right, of the myelin sheath, it becomes white. So then it's white matter, okay? Remember that we're talking about the central nervous system, okay? Okay, so we have lots